Can the online audience hear me now? Could someone online say if he's hear me or not? As our visualization of the complete Mihai project, we have different parts here. How you can see the microbes is in the all parts. We have microbes in the plants, we have microbes in the phonics, the micro full cells, and also in the MEC. But we also, what we plan basically in Mihai is to produce plants to have clean energy and also have a circularity inside the system. I will go later on detail on this. But yes, who we are inside Mihai. So inside Mihai, we have Kaluven, that's the university in Belgium. We have University of Southampton. We have the University of West Bristol. We have an institute in Spain, a Council of Superior Investigations. We have Biofaction that is most responsible for marketing. And we have ourselves here in Sony. So our main responsibilities inside the Mihai project as Sony team and CSL team, we have a big package that it is about the data analysis that we at least promise it to do some AI and models and algorithm related to Mihai. And we also have technology integrated that this main focus is about sensors for the system and how to monitor the system, how can we control the system, how can we automatize the system. So the first part that it's about data analysis, we have mainly two blocks inside our package in Sony. That is system integration, that is related to modeling each part of the system, and you're looking of your optimization and integration of the parts that our partners do. And we also having the microorganism part that we want to study how the biofilm in the MFC are formatted and what biodiversity or what is the genetic we have from microorganisms inside the Mihai system. So just to make a little bit of overview what we have been done until now. This is the first model we did for Mihai. It's a general model to show the different parts of the system. For example, as input, we have information from our partners in Southampton about the MFCs, that is the responsible to produce energy. For those that don't have a biological background, just to understand a little bit about the MFCs, you have like microorganisms, mostly bacteria, that you put some substrate that in case of Mihai, we want to use wastewater or urine. And these microorganisms, they clean the substrate. And during this clean process, they produce energy. It is basically this is alive batteries. We can say like that. Then you basically input the substrate here, for example, wastewater in the anode, and you have this transmission from the cathode, and you have in this side, you say clean water with some microorganism, and you also have the energy production. The idea is to have this cathode that is the clean water plus the microorganism being put in the hydroponic, so you use less artificial nutrition and you can produce plants, and also you can use this energy from produced from here to illuminate the plants and make the plants grow. So in this model, we get all the information that we have the MFCs, how much substrate we can input, how much energy is produced, how much nutrition we have here. to be put in the hydroponic, and also get the information from hydroponic, how much plants we can produce, how is the nutrition of this plant, how much artificial nutrition we need to complement from the nutrition we have from the MFCs. And in the end, what we have more or less are numbers about how much energy are produced, how much nutrition solution are produced, how much of this nutrition solution have of nutrition for the plants, and here how much the plants taking from the air from CO2, what's the nutrition of these plants that can be eaten, of course, and how much exudates are produced. These exudates are nutritions that the plants don't take from the root, but they change the chemical format of the nutrition, and this is available in the water and can be later used for other microorganisms. And also, we know more or less how much energy needs to be consumed for the plants to during the cycle, and if this is balanced with the energy that we can produce in the MFCs. So this is a general model that we create in the first year. So beside that, what we want to do here in Sony as a package, we want to have some samples for the MFCs, for the biofilm, for the bacteria that produce the energy, and we want to modeling how these bacteria are growing in the to form the energy, and if you exchange the growth of this bacteria or the species of this bacteria, if you can produce more or less energy. This is another target that we have in house. And the target we have for the microorganisms is to collect the microorganisms that we have here in the MFCs and all the system and see the biodiversity. For example, if you take the wastewater to produce energy and you want to clean this to after that produce a food or being or human urine and produce food, you need to know what kind of microorganisms are there. If you have some wizard disease or something like that, if you really clean the water, and also which kind of microorganism will be inside the system, if they are good or if they are bad for the plants, and if it's possible to produce plants with these microorganisms. So for our second part that here in Sony, we also have the technology integration that is mainly related to sensors. And I divide here in two kinds of sensors that we are using right now. That is digital image that we are using cameras to see how the plants are growing and how is the nutrition in the plants, if the plants are healthy or not, if you need to change the nutrition. And we also have IoT sensors that is most related with the water quality inside the MFCs and the hydroponics. And also the sensors are using to measure the energy that are producing inside the MFCs. So in this kind of package, this is downstairs in our lab. I don't know if you saw during the first test that we are doing, the first try, is the hydroponic system with the light, as I showed before the model. This is kind of IoT system. This was a company made, was the first one we bought it, but nowadays we have different model that we optimize for our case. And what we want to do, for example, with the cameras, we are using the Raspberry Pi that is a Sony sensor camera, IMAX 708. And we take pictures of the plants that are there. And what we can do, we can program these pictures. We can create a mask and make a black background, and we can extract the green part of the plant. And with this green, we can estimate if the plants are not health or health, if they have enough nutrition or if they don't look fine, and then we can investigate why they don't look fine. And also we can estimate the growth of the plants, how much it is the how much green is becoming, how much these plants are growing over time. I did here the most easiest way as possible to explain, but we have a lot of complexity to see how much the plants are growing, because you know that the plants growing overlapping leaves, So just taking pictures from the top maybe will be not enough to estimate how much the plants are growing. Then what we are testing in the next round is to install cameras in different positions and angles. If we can get better information than the recommend, okay, you should install the camera in this position, and you can get a estimate of, or you can get a software or modeling that you can see how much the plants are growing when you should harvest it. And in our experiment, we are mostly using basil, not just because it's used worldwide to do pesto, but because it's like is one of the easiest plants to grow in hydroponics, at least. So, as mi-high target, we have like the below part, the biggest target we have, we want to produce a clean energy. We want to have this bio battery that we produce energy and can substitute the energy that we use nowadays, that we need to put fire and we need to fall like fools to produce energy. Then we want to have like clean energy. In the second layer of the pyramid, we want also to have the use of the resource as better as possible, because nowadays in hydroponic you put 5 times more nutrition that the plants needed, expecting that the plants are growing fine. And we have like an artificial environmental, because you took out the soil from the plants and you just gave water. Then you need to give as much nutrition as possible so the plants don't be stressed and they can grow. So what we want to do, we want to more or less have also some microorganisms there that keep the plants not in the optimal condition, but in the condition that they are kind of a little bit stressed, but they still producing, because this a little bit stress can give more tasting also to the plants. And in the top of the pyramid, of course, we want to be innovated, because we want to have a circularity, and we want to have a self-sustainable system that we don't need to power with the external energy. We will have the energy inside, we have the plants inside, we have the microorganisms inside. And as us as a human being, we will interact with the system more or less just like controlling the system, and also harvesting, doing like minimal procedures, but it still need us. But the system needs to have a kind of circularity that you don't need all the time change all the parts. And as numbers, I put some numbers that, of course, this is like our dream targets. We want to have 24/7 monitoring the system and the plants, of course. We want to have 100% of all the energy that we have inside the system done by the system. We want to produce our own energy inside the system. And we want to, of course, use less fertilizers to produce plants. We want to use less nitrogen as possible. And we want also to have a rhizosphere for the hydroponics. Rhizosphere, is a term that means microorganisms that are around the roots and help the plants grow. For example, help the plants get the nutrition, help the plants grow the roots, help the plants stay healthy. And one thing that is also really nice inside hydroponics that I didn't put here because nowadays already it's like this. In the hydroponic systems, you don't use pesticides. You don't use chemicals to control insects or to control disease. Because I still have a clean system, you don't have like, you have an environmental control system, you should not have disease because you have sensors, you have the system that you control yourself with the temperature, pH, and the humidity. Normally, you don't have disease. Then you have like a healthy plant that is pesticide free, and our target here should test it as close as the plants that you harvest in the soil. This is the idea. And what should be the deploy scenarios? Since we start the project, we discuss a lot where we will use Mihai. If we will use Mihai directly in the farm, we will use Mihai in the city. But some scenarios that you could see Mihai is, for example, in urban settings, as we have in our rooftop here, that we do experiments with the garden in Paris, because should be a system that could be planned to be small ones, but you also could have Mihai in a big scale, and you could have it in an indoor farm. For example, here I put a picture of a farm in the Emirates. It is a case that you don't have a lot of water available for you, and you have an indoor farm. And then, if you have a system that you can clean the water and reuse the water, or even use it water for buildings, for example, wastewater for a building, will be really optimal situation. And of course, if the world end, we pretend to be there too, because this is our idea. We want to see what's the worst scenario ever. At least we want to address climatic change. For many people, don't look like the end of the world, but I can prove that is. uh, Today, as not so Brazilian niceness and hopeness, I am want to show you what's the perspectives for the climatical change. I hope don't scare you guys, or maybe yes. But we have a lot of cases. We have desertification, we have increase of temperature, floods, deforestation, disease outbreaks, for example, Corona. I don't know if you remember. No political system, climatical migration, soil degradation, collapse of food production, water acidification, end of limited resource, and loss of biodiversity.
What is non-political?
Yeah, no political system. I will put it here. ChatGPT told me this. Then I don't know if you trust him. I trust sometimes. And no political systems. You mean in case you have like collapse of food production and you don't trust the system anymore, the political system will collapse it too. We didn't arrive here now, but it's ChatGPT see this as possible, and I will prove now scientifical that some things are already happening. For example, you saw in the news that we had flooding in the Canaries last year, and this was a tragical thing that also happened in Brazil. The same thing. We have floods in Brazil, in south. Was like the capital of Rio Grande do Sul. It's a state in Brazil. Was completely floating. People lost their house. Was really, really, really sad. This also happened last year in November. And in case you don't believe in ChatGPT that you should go and look for papers. Here are some papers. Here, it's some scenarios in case of the climatical change continue in the direction that we are, because we are not doing something to mitigate it. Then we have some scenarios. For example, if we're still using the pesticides the way we use, we will have pesticide pollution. We will have, if you still use the water way we use, we will have water scarcity, and so on. For the nutrition, we will have NMP pollution because we using the chemical way. We will have soil degradation. We will have problems how to produce food to feed the population. And of course, we will have loss in biodiversity, because nowadays we most eat for oh my God, four crops. We have four big crops, that is corn, wheat, soybean. I forget the last one, I think is rice. These four crops are the major base of everybody food. And then we are lost in biodiversity because we are not producing, we are not having look and breeding the other crops. And for those that want to go deep in and be more realistic than ChatGPT, this is a paper published in Science last year, and it is climatical change exacerbation, the environmental impact of agriculture. And I want also collaborate with another paper that was published this year in February that was saying solving climatic change requires changing our food systems. So this is a mainly conclusions from the paper. The unsustainability of our current course and the urgency for actions to change our food system are undeniable. I hope that is clear. Our survival that of no human in animals, ecosystem, and planet depends on recognition, the interconnectedness of the life and our mindfulness in the choices we made it. We will not success on the issue of climatic change unless we change the way that we produce and consume food. Then, guys, the thing is serious because 27 researchers work together around the world to write this paper and tell people, let's pay attention because the scenario is not the best. So, we as Mihai, how we will save us from the end of the world? This was my pitch for today. So the answer is spica, and I want to speak a little bit about it. So spica is described as deep tech in ancient biodiversity. It is a system that we envision that we can have in the most catastrophic scenario, I think, like the end of the world, we still alive, some humans, and we need to produce food. How could you produce this food with the high technology? That is spica. So I will go a little bit in detail of the parts and how Spica was created. We imagine that in the end of the world, we still have some pollinators and some insects, and this kind of animals need to have an option or a place that they can uh, build their house, that they can have the pollination. They need to have free roads to fly around, and this spica will be this kind of system. Also, in spica will be possible that some kind of parasite or some kind of fungi or microorganism that will be there, create after our pollution, can grow around the spica and can be integrating the system, not as enemy, but as like a helper for the system. We also have, of course, the parts in the system that we building and we imagine ourselves. We have the hydroponics to produce food, to produce at least to grow plants. We have inside the hydroponic the microorganisms and all this collaboration for plant production. But we also have outside the spica biopanels that will cap the airborne particles and will have like some microorganisms, algae, corals, or something else that came from the air and can grow there and can help it to clean the air. We also have the MFCs that will be responsible to create all the energy that spica will need to produce food in the hydroponics, and also have the microorganisms inside. And in the below part of spica, we imagine that the soil really changed. We don't have like soil anymore, that we actually have like slime. That's a kind of mix of microorganisms and particles that need to kind of eat our electronic waste and some kind bring our soil back. And we have, of course, sensors that we will control all the systems, all the plants when we need to harvest. And we have us, maybe not as we are nowadays, but it's still us, like in thinking and going to the systems and being in this case as cleaner and harvesting the food, seeing what microorganisms should be growing there, what microorganisms should be inoculated, what microorganisms should be taken out. This is the futuristic way we envision spica there. But how close we are to have a spica in the real world nowadays? So I have good news for you. If the apocalypse arrives, we have the spica at least. If the spica will be the solution, I will let the future know, but like we have the spica. And why we have the spica? The spica concept and the idea came from an invitation that Rachel Armstrong, that is the responsible for our project, she got an invitation from Triali in Milano to do an art exposition about the microorganisms and futuristic way to envision this microorganism. This invitation was got in October last year. She got this as idea to use our project Mihai to envision the future and the microorganisms. So the exposition actually called inequalities, and they have many different areas. They have our also how they envision the new big centers, how they envision the new cities, and our expositions is inside this exhibition with the bacteria nodes to biotic architecture. And this is how the Milano Triale looks like. If you have the opportunity to visit, it's really beautiful. And the from October on started the idealization of how the spike will look like. So this idealization started mostly for Rachel team in Belgium, because they are architects, they have better imagination and background to see how can we look like this piece of art. Then the main ideas for Spica was to be something like medieval, but also something monster because it's the end of the world, of course. And of course, it be related to microbials in somehow, like be survival, be some kind of virus, but also be some kind of arachnidio, and also being inspired by some church. And also, of course, it be defensive, because if the end of world, where you want to produce your food, but you don't want everybody harvest it. So this is how the spica look like. It's a 6 meter installation. It's look like really like fortress, but it's also it's an entire ecosystem. Uh, More or less what was the timeline we had. We got the official invitation in October last year. We have a lot of discussion meeting to see how we will be the spica and how spica will look like, the pieces of spica. Then in January, we have the visualization of how this look like, and we start to build the parts, the hydroponic, the MFCs, the panels, the sensors, so on. And we finish spica in May to be totally complete and construction inside the Milano Triale. And the open exposition was also in May. And now from May to November 25, maybe this extend a little bit, we need to go there and do maintenance, because this spica is a alive piece of art. We have plants, we have microorganisms, then we always need to be there taking care of the spica. So I want to go in detail of each piece of spica. First of all, the architecture, how the spica look like. It's really like a Lego. This was constructed bit by bit in this sequence, because we need to have all the pieces inside, and the end we need to construct everything around that people cannot enter inside. That is protected. Then first the tower are built with the main parts in the middle that we can put it inside the hydroponic tower. Then in the second part, we also put here the legs and the MFCs inside, and the biopanels came. And in the last part came the parasite, the spikes, and also the sensors. But I will show pictures for you. So this was how the construction looks like there in Milano. The steps here, you see the tower with the 4 legs, and here you see the main part in the middle to already put the hydroponic part, and you see the parasita. These are the spikes that was put in later on around, and also the other legs that was put in the middle of the spike. About the hydroponic, we also have like a process. We need to buy the plants, select this was like before we started Spica, we bought it the hydroponic tower and we did some experiment what kind of plants can survive inside a museum with like minimal care, and we decided for these plants, and then we built in the tower, and was I think it was the second step. It was the spica tower first, and then we built the hydroponic tower and we put it in the top of spica. Then we also have like the process for the biopanels. Uh, we first monitor the water that we had in the park next to the 3LA. We saw that we have a lot of algae production and probably microorganisms there. We collect the samples and we check the pH. After that, we have this was also a puzzle, was like a pieces of the biopanels. The biopanels are made from two materials, was cement, ceramic, and wood. And we inoculate the biopanels with this water. And after that, we mounted it in around the spike. For the MFCs was the same. We brought all the MFCs to the Polytechnic of Milano. We mounted there first. We did some trials, and after the MFCs was working fine, we put it then inside the spiker here. For the sensors, that was Sony responsibility actually. We had the sensors for the hydroponic water, and we also have cameras. Then after Spica was completely mounted, Peter and myself, we went there and we install the sensors here inside in the water tank. Then we can control pH, electroconductivity, oxygenation, and also CR2 inside the spica. We also have cameras around the biopanels to see how much microorganisms are growing in the biopanels and how much we have a camera in the hydroponic to see if the plants are doing fine or not. We also have inside the MiHi project, we have people that are responsible just to marketing out what we are doing. And this is really like also a hard process because they need to visualize how Sparkle look like, building some pictures and also do some press release. We even have a Sparkle website that you can enter there and see how Spica look like and all the history about it. So I want to thank you very much, everybody, that it's part of the project. It's a lot of people between PAs, PhD students, postdocs, master students, and interns. These are some people that we are together there in Milano. People from Madrid, people from these are the girls from Belgium that was responsible for all the architecture part. These are the professor responsible for the clinical parts. Here also we had people from market and divulgation. We had the postdoc from architecture. This is Rachel that is responsible for the project and was invited to do the exposition. And yes, this is all of us there in the open day. Was really nice day that we saw the Spica completely building. But I also put here Marcos that was not there in the open day, and Leonor and David that was helping us since the beginning with the camera and all the project. And yeah, that's all. I also have a QR code here if you want to see more, and you can scan. The idea of my presentation was like to show what the project is about. It scare you a little bit with the climatical change, then maybe you can sleep on this and start to do something also to change. We can do in our day-based life, but we can also do as research, of course. Uh, and also to show how amazing is Spica. You can visit the web page, you can visit it in Milano, and just a less information that I also listened in a conference 1 month ago. We can really do simple things change the world and to mitigate climatic change. For example, sometimes we enter to ChatGPT and we ask him to produce a photo of ourselves doing something fun, and the energy that consumes just to produce that photo, you can produce one plant of lettuce. Then you need to, in your daily life, you can make some choice that you can save energy, and indirectly you can save, of course, the planet. Thank you very much. Yeah, sorry, don't just scare everybody.
No, no, no. Thank you very much, Lenon. The questions? Maybe I can start with one.
Okay.
If you go back to model figure,
Wait. Oh, yes. Introduce. Yeah.
So I understand that in urine, for example, you would get some nitrogen.
Yeah.
So the N is okay, but where does the P and K come from? Also, it's naturally present in urine.
Yeah, no, this PK will come if you use wastewater.
Okay.
Then depend what kind of substrate you put it in the system, with different kind of catalyte we will get it.
Okay.
Yeah.
Any other questions?
How is Spica going?
Yeah, it's more challenge that we thought it, because you have a piece of art that is alive inside a museum. It is really challenge because the plants are not in the best environmental, and we just have one day a week to go there and to do some checking. But so far it's doing good. But yeah, it's really challenging. I can tell you, we are spending a lot of money like to keep this running, because it's the first prototype for the project, and it's directly inside the museum. Then this is challenge, yeah. And no one from our collaboration have a lab in Milano. Then we are collaborate with another university that is taking care of our work. Then we need to explain them what our work it is. Then yeah. Yeah, you should visit this park. Important to go Milano.
Okay, thank you. If there are no further questions, I think we can close here.
Thank you.
Thank you.
Thank you. Ciao.